Hey guys, we're going to pick up where we left off and we're going to start talking about the cell-mediated immune response. Remember, this is the second arm of adaptive immunity, which is specific immunity. So we're going to go ahead and jump on in. Okay, so the cell-mediated line of adaptive immunity is different than the humoral line of cellular immunity for one big reason, and that is what happened to the antigen. So in humoral immunity with our B cells, remember, humoral immunity will occur if a free floating antigen comes in contact with a naive B cell, and the naive B cell takes in that antigen and presents it. Or it could happen if another antigen presenting cell like a macrophage or a dendritic cell engulfs an antigen and then presents it. The key thing here is that the cell is taking in the antigen voluntarily and it is presenting that antigen externally. So we call these exogenous antigens. So these are antigens that were floating around in the humoral fluid, and then the cell presents them. So the cells were never actually infected, right, with the antigen. With cell-mediated immune response, this is the direct response that's going to occur whenever a cell gets infected by the antigen. So if a macrophage, is not purposely engulfing the antigen and forming a phagosome and killing the antigen, but instead, let's say a virus infects a macrophage, that macrophage is going to be able to present some of that viral antigen on the outside of it. It's kind of a cry out for help, like, help, I'm infected. When that happens and it presents its antigen, we call those endogenous antigens because they are created inside the cell due to an infection. And so those antigens are then going to provoke what we call cell-mediated immune response. So we're going to talk about some key players, and then at the end, we'll kind of put it all together, okay? So cell-mediated immune response is 100% dealing with T cells. So in humoral immunity, we talked about B cells presenting antigens to helper T cells, and those help activate the B cells. Well, with cell-mediated immune response, we're strictly talking T cells here. So T cells are going to provide defense against intracellular antigens. They're really, really good at activating macrophages. They're really good at getting other T cells fired up. They are really your regulators of the immune response, and they also help mediate inflammation. So adaptive immunity, the cell-mediated side, while it is very specific, it still is intertwined with innate immunity. It's going to increase those macrophages. It's going to enhance inflammation. All of those things are still going to come into play, okay? So there are two major types of T cells out there. There's T cells that are named for their surface proteins or their receptors. So some T cells have a receptor on it called a CD4 receptor, and other T cells have a receptor on it called a CD8 receptor. CD4 T cells are referred to as helper T cells. CD8 T cells are referred to as cytotoxic T cells. So when a CD4 cell is naive, we say that it's non-activated. Once it is activated, it becomes a T helper cell. So a CD4 cell is what we talked about when we were talking about humoral immunity, and when like an antigen is presented by the macrophage to a CD4 cell, and that macrophage antigen can be recognized by the CD4 cell, then it activates the CD4 cell to become a T helper cell. CD8 cells, when they are activated, they become cytotoxic T cells, which are really good at destroying antigens. CD4 cells never destroy anything. All their job is to activate other cells to do their job. T helper cells don't sound like they play a big role, but they do. If you don't have a T helper cell, you don't have immune response. So T helper cells are vital, but they're not the ones actually doing the killing. Now, there are some other types of T cells that we're going to talk about as well. There's T regulatory cells and there are T memory cells. The T memory cells are much like the B memory cells. They're going to be stored away later in case there's a secondary immune response that occurs. T regulatory cells are really important because what they do is they tell the immune system to slow its brakes, right? To pump the brakes whenever the infection is waning or going away. The T regulatory cells tell the immune response to just chill out for a minute and not to make so many active immune cells. And this is so that the cells don't go rogue and start attacking body cells on accident. And so these T regulatory cells are going to be important for that reason. So the main comparison between humoral adaptive immunity and cell-mediated immunity is really and truly the target, right? So the bacteria and molecules in extracellular environments, okay, these exogenous antigens, are going to be the target of humoral response, while the endogenous antigens are going to be the target of the cell-mediated response. Okay, so T cells of the cell-mediated response, like I said, only recognize and respond to cells that are going to be processing an antigen and displayed on the surface of the cell's body. So these are cells infected by viruses or bacteria. Sometimes these could be abnormal cancer cells, or they could be cells that are infused or have transplanted foreign tissue on them. Okay, and so because they'll be creating a new type of protein that the body has never seen before. So just to have this word written down, okay, these are endogenous endogenous proteins, while, clear that out there, while humoral are exogenous proteins. Okay, so just having those two words is going to be helpful. Oh, goodness. Of course, it's going to keep doing this to me. My brain has to turn on. Okay. So let's talk first about how the T cell recognizes the antigen. So remember that T cells have to be immunocompetent, which means that they have to be able to recognize self versus non-self. So when a T cell comes in encounter with an antigen presenting cell, the T cell has to simultaneously be able to recognize the non-self portion or the antigen, and at the same time recognize the self portion. That self portion is called the MHC protein, or the major histocompatibility protein, or histocompatibility complex protein of the cell body. So MHC proteins come in two varieties. You have MHC1 proteins, which any nucleated cell in your body has it. If it is a cell with a nucleus, that cell in your body has an MHC1 protein. And that MHC1 protein's job is to display self-antigens. So this is a body cell, and on here is this MHC1. And that MHC1 should have a self-antigen on it. Okay, so that's really important because if you don't have a self-antigen on that MHC1 protein, then the cells know that that's an invader, okay? Now, there's only a couple cells in the body that are going to display what we call MHC2 proteins. These would be any of your antigen-presenting cells. So your dendritic cells, your macrophages, and your B cells can all have MHC class 2 proteins. So they're still going to have the MHC1 proteins on them with the self-antigen. Sorry. But they're also going to have an MHC2 protein, which is capable of displaying a foreign antigen. And that's what makes them unique, right, to the body, so that they can display this foreign antigen to a T cell. So all nucleated cells in your body have MHC1 proteins. And then only your APCs, your dendritic macrophages and B cells, have MHC2 proteins. Okay, so let's talk about these MHC1 proteins. MHC1 proteins are going to make endogenous antigens, which are antigens made inside the body. Okay, and so these again are self-antigens, and that's how it should be in a normal cell. Okay, in a non-self antigen, you're also going to have endogenous proteins, but they're not going to be self, right? These are going to be foreign. So if I have a bacteria cell, okay, that's in my body, it's also going to have an MHC1, but it's going to have an antigen that doesn't look like ourself. In an infected or an abnormal cell, something really unique happens. The MHC1 protein is not only going to have the self protein on it right here, but if a virus gets inside of this cell, that virus is going to create a protein as well. And that protein is going to go hop up on our self protein, okay, and it's going to make it different. So this infected cell is uniquely going to have an MHC1 protein with self antigen and non-self antigen. This is different than when a cell is not infected. So if a cell engulfs, right, if it engulfs the antigen, it's going to have the self MHC1 protein, but then it's going to have the non-self protein on an MHC2 protein, okay? So right here, this right here is what is going to trigger, okay, the CD8 cells. This self protein, along with this non-self protein attached, is going to tell CD8 cells these cells are infected and they need to be destroyed. Okay, and so these CD8 cells are going to come in contact and they're going to activate themselves into what we call cytotoxic T cells. Okay, and this tells the CD8 cells that there's a microorganism like a virus or a bacterium hidden within the cell, and the cell needs to be destroyed. So this is kind of showing you a better version of what I just talked about. These are the endogenous antigens. So this is what your normal cells would do. So in your normal cells, you would have your own body creating a class 1 MHC protein, okay, with a self-antigen on it. But when a endogenous antigen comes in, so like this might be a virus or a bacterium or cancer. Normally it's virus and cancer, okay? The cell is going to, the good cell, your body cell here, is going to break up these little antigens, and they're going to be processed and they're going to be displayed on the outside of the cell, okay? And that's going to tell our body, hey, there is a viral component in here or a cancerous component in here that's bad, okay? Because it shouldn't be on that class 1 protein if our body had engulfed it. This tells us that this cell is actually infected, okay? Now, MHC class 2 proteins, which we talked about yesterday when we talked about humoral immunity, those are going to bind with a couple of different things. They're either going to bind with fragments of free-floating antigens or fragments that have been engulfed and broken down in a phagolysosome. So this would be not when the cell is infected, but when the cell has purposefully taken in the antigen. Okay, so this is showing you that mechanism. So in this cell, okay, we have actually engulfed the antigen. That antigen is going to fuse with the lysosome and form a phagolysosome. And all these little pieces are going to break down. The nucleus, not the nucleus, the endoplasmic reticulum here is going to produce MHC class 2 proteins that are going to make a vesicle and fuse with the little pieces left over from the breaking down of that antigen, and that's going to be presented on the outside. This cell is not infected, so this cell does not need to be destroyed, and so that's why this antigen is produced and displayed on the class 2 receptor. So this just tells CD4 cells, not CD8 cells, CD4 cells, hey, I've encountered an antigen, I've destroyed it, but I want you to know that there's other antigens out there that are probably like it. So it's more of a signal or a warning. not a distress sign like the display on the class 1 MHC proteins. Okay. So in order for T cells to become activated, antigen-presenting cells are going to migrate to the lymph nodes, okay, and they're going to present the antigen that they found to the T cells. So T cell activation is a two-step process. The first thing that's going to happen is the antigen is going to bind, and then the antigen actually has to do something called co-stimulation. Co-stimulation is an important step because if the antigen binds, okay, but it's not co-stimulated, then the T cell will actually shut off and it won't turn into a cytotoxic T cell or to a helper T cell. It'll just go along its business and keep roaming around and looking for a new antigen. But if it binds and co-stimulation occurs, then that's the trigger to turn the T cell from a naive cell into either a cytotoxic T cell or into a helper T cell. So dendritic cells are able to obtain other cells' endogenous antigens, again, by engulfing them, okay, and then importing those antigens onto the surface of the cell. And they can display those antigens on both class 1 and class 2 MHC molecules. Now, these T cell receptors that recognize the non-self complex is going to be linked to a whole bunch of pathways. But essentially what those pathways are going to do is they are going to recognize that that is a non-self antigen, and it's going to trigger CD4 and CD8 cells to activate themselves. Okay, this antigen binding is going to stimulate the T cell, but again, it's not going to activate until co-stimulation occurs. So co-stimulation occurs whenever two more antigens or two more receptors link up, okay? Dendritic cells, there we go. Okay, so this is what it's going to look like. So here is your dendritic cell, which has gotten a viral antigen inside of it. So it's engulfed this antigen. It has not been infected by it, okay? It's engulfed it. It's going to present that antigen on the class 2 MHC protein. Okay, it's going to go and it's going to link up to a CD4 cell. The CD4 cell is going to recognize that this is a foreign antigen, okay, and then it's going to have co-stimulation, which we see here in purple. That second protein, okay, that links up is then going to tell the CD4 cell to start making more copies of itself. So this is a T helper cell. Some of these T helper cells are going to become memory cells, just like the B cells. Remember when they made copies of themselves, they became memory cells. So some of these T helper cells are going to become memory cells, and others of these T helper cells are going to become activated effector T helper cells. These effector T helper cells then are going to release a series of chemicals, interleukins, right, which are then going to activate other T cells like the CD8 cells, or they're going to activate B cells and allow them to divide, like we learned about in humoral immunity. Okay, so that co-stimulation that's occurring, okay, is a second binding of receptors. So on dendritic cells and macrophages, on their surface, they have something called B7. And then on the T cell, they have a receptor called CD28. And that's what we see here. Okay, this is the CD28 receptor, and it's attaching to the B7 basically in order to make co-stimulation occur. Now, when this co-stimulation happens, cytokines, specifically interleukin 1 and 2, okay, from the antigen-presenting cell or from other T cells are going to trigger that T cell to replicate. So these two interleukins are going to be important for allowing this step right here of the T cell differentiating and turning into more cells or proliferating into more cells. Okay. Now, if there is no co-stimulation, if this little secondary protein doesn't match up, okay, then anergy occurs. And anergy is basically deactivation. So the T cell, okay, sees that antigen and becomes tolerant to it and says, oh, well, I recognize this as non-self, but actually it's all right. I'm not going to worry about it. and the T cell will inactivate. So this makes the T cell not divide into memory cells and into other helper T cells. And so therefore it's not going to secrete cytokines. Therefore it's not going to provoke cytotoxic T cells or B cells to continue on with the immune response. T cells that are activated, however, are going to get bigger. They're going to divide and they're going to produce clones. And then again, some of them are going to differentiate into helper T cells and others into memory T cells. Now, primary T cell response will peak within a week. So the first time, right, we have that we encounter the foreign antigen, okay, you're going to have about a lag period of about 3 to 6 days. And then within a week, we're going to have peak response. After a week, T cells are going to start dying. Okay, we call this apoptosis. And this is going to occur between day 7 and day 30 after the immune response begins. And this is basically going to wane the amount of antigen in the body, because if T cells are declining, okay, not wane the amount of antigen, wane the amount of antibodies, sorry. Because if T cells are declining, then it's not affecting the B cells, which means that they are not going to be making a lot of antibody. This is typically in response to antigens declining in the body. So as the antigens are being destroyed and there's less and less of them, then those T cells are going to start destroying themselves. The benefit of this, okay, is because anytime a T cell is activated, it's a hazard to the body because it could accidentally kill body cells. And so if there's no need to have all these active T cells running around, then the body cells are not going, then the body cells don't want the T cells around. And so programmed cell death is going to start to occur, and we call that apoptosis. Now, some of these T cells, remember I said, become memory T cells. These guys are going to remain forever. They're not going around destroying. They're not going around activating other cells. The memory T cells hang around inactive until you get a secondary immune response. Okay. So those activated T cells, remember, are going to secrete cytokines. Cytokines help with differentiation. So if a helper T cell is secreting the cytokine, then it's going to tell the B cells from humoral immunity to differentiate into memory B cells and into effector B cells or plasma B cells, okay, to respond to the immune system. Cytokines include interleukins like IL-1, IL-2, IL-5, IL-6, IL-10. There's a lot of them, okay? And then also those interferons. Remember, interferons were what is secreted whenever a healthy cell is infected by a viral cell. So remember, the body secretes interferons to go signal other neighboring cells, right, to block viral entry. And so those interferons are really good things to be secreted. Now, in terms of cytokines, IL-2 or interleukin-2, okay, is one of the key factors that act on the cells that release it and other T cells. So what the IL-2 does is it encourages the T cells to divide very, very quickly. Okay, and we use IL-2 actually in therapeutic mechanisms to treat melanoma, which is skin cancer, and also other kidney cancers. So we can actually isolate this cytokine and do that. Other cytokines help amplify your innate response. So for example, they'll go in and they'll activate more macrophages, or they'll go in and they will enhance inflammatory response. Okay, and these other interleukins will also help other parts of the adaptive immune response. So maybe they go and enhance the B cells, or they'll go and enhance the CD8 cytotoxic T cells. So these helper T cells, again, play a central role because once the primed T cell is presented the antigen, okay, that T cell becomes active, the helper T cell, and it activates other T cells like the CD8 T cells and activates the B cells. It makes them proliferate. It also helps innate immunity by activating macrophages and recruiting other immune cells. So they really are the kind of key players to make everything else work. Now, remember that helper T cells interact directly with the B cells, displaying any kind of antigen on the MHC2 receptors. These cause the T cells, whenever the antigen on the B cell comes in contact with the T helper cell and it activates, it stimulates the B cells to divide rapidly. And then those B cells then again can create antibodies. Remember, because those are plasma B cells. Okay. So B cells may be activated sometimes without helper T cells, but it's not as common. Most of them require T helper cells to activate the B cells. So this is just kind of peeking back into humoral immunity here. Okay, here we have our activated helper T cell. This was our B cell, and our B cell presented the antigen. The T cell becomes activated when it recognizes this as a foreign antigen. It produces cytokines, and those cytokines are then going to activate the activate the B cell, and that B cell can then differentiate and proliferate into plasma B cells and memory B cells. Okay. Helper T cells also do the same thing for dendritic cells. They let the dendritic cells express co-stimulatory molecules required for CD8 cell activation. So when a dendritic cell is infected by a virus, okay, it can actually use a helper T cell to help co-stimulate it in order to respond with a CD8 cell, to turn a CD8 cell into a cytotoxic T cell. So this is what we see going on here. So here we have a dendritic cell, okay, and it's got a viral antigen in it, and it is presenting this to the CD8 T cell. But when it presents it, the CD8 T cell does not become activated. It'll only become activated when the helper T cell also binds to the dendritic cell. That is co-stimulation. So now the helper T cell will then produce cytokines, which tell the CD8 cell to start dividing in response, okay, to this dendritic cell. So again, this CD8 cell is a cytotoxic T cell and can directly begin killing these bad cells that have the antigen infected inside of them. So CD8 cells or cytotoxic T cells directly attack the cells. And they do this by secreting two main things. The first thing is they're going to secrete something called perforin, which is going to puncture a hole inside the cell's membrane. And then it's going to secrete something called granulozyme. And that granulozyme is then going to get inside the cell of the cell that we're trying to destroy. And it's going to cause the cell to do apoptosis or programmed cell death. There's a whole big mechanism that happens, but basically the granulozyme is going to affect something inside the cell called the Bax protein. And that protein then attaches itself to the mitochondria, and it splits open the mitochondria, and the mitochondria releases something called cytochrome C. And then that cytochrome C then is going to set off a series of proteins, which are going to basically break down the cell from the inside, and all of the inside of the cell is basically going to spill out. So you don't need to know every little thing that's going on inside the cell for apoptosis, but I do want you to understand that that's the mechanism in which cytotoxic T cells work. So cytotoxic T cells are only going to kill virus-infected cells, cells that have been infected by parasites or bacteria, cancerous cells, and then any kind of foreign cells that come into the body that are mismatched due to transfusions and transplants. So these cytotoxic T cells bind to a self-non-self complex. So they're going to bind to that MHC1 protein that has an MHC, or that has an antigen presenting foreign antigen on it. The lethal hit, again, that causes the cytotoxic T cells, sorry, I got ahead of myself earlier, is when perforins and granzymes are going to actually cause the cell to undergo apoptosis. So I just explained that. Perforins create the hole, and then granzymes enter the cell, stimulate a whole series of things going on, and then the cell divides. Okay? All right, so this is kind of what it looks like. So your cytotoxic T cell here is going to match up with the target cell. So this would be the virus-infected cell, for example. And when it does, it punctures a hole, okay, into the virus-infected cell here, which is this yellow cell. And then all of these little granzymes, which are proteins that are created by this cytotoxic T cell, are going to be secreted inside of the cell. Oops, sorry about that. Okay, so that is what's going on in adaptive immunity. It's T cell mediated immunity response. Just to kind of jump back into innate immunity really quick, because all of this is so intertwined. You remember those natural killer cells? Natural killer cells were not adaptive. They're not worrying about specific antigens like adaptive immunity is. But remember, they're going around and they're killing things in the innate system. Anything that lacks a class 1 MHC antigen, anything that an antibody has coded, or anything that has a different marker on the outside of the cell that tells the natural killer cell it's not supposed to be there, right? These cells, the reason why I want to refer back to them is because they use the exact same mechanism to kill their cells as cytotoxic T cells do. Okay, so they are going to actually use perforin and granzymes just like cytotoxic T cells do. The difference is that cytotoxic T cells are looking for a specific antigen, okay, and they're not just bumping into these random antigens that are moving around in the body. Okay, remember I said that regulatory T cells also are going to play a role in this immune complex. So regulatory T cells are going to put the brakes on the immune system, okay, whenever there is a lack in antigen. So as the antigen or the invader starts to die off in the body, regulatory T cells, okay, get amped up, okay, and they create something called inhibitory cytokines. These are little chemicals that go to the cytotoxic T cells and they prevent them from continuing to kill cells in the body. And this is really important because it prevents autoimmune reactions in the body. Sometimes our immune system doesn't do what it's supposed to do, and we get diseases like vitiligo or like MS. MS is like the disease in which the body attacks the myelin sheath, right, surrounding the neurons. And that's really bad because it slows down motor control. Diabetes type 1 is also an autoimmune reaction. Basically, your body starts killing the cells that secrete insulin. Sorry, my brain just stopped for 5 minutes there. Okay, so those regulatory cells are going to put the brakes. So this is kind of what we just talked about, and this is what I would probably jump into putting on my flow map if I were you. Okay, so you did a humoral immune response, right? Humoral immunity, which we see here in blue. So we did this yesterday, kind of. So we said like a free antigen is going to bind to a B cell. Those B cells are going to come in contact with with helper T cells, which are going to cause the B cell to become activated and produce plasma cells and memory B cells. And those plasma cells then are going to secrete antibodies, right? So flipping over to the cell-mediated side, okay, you're going to want to not talk about free antigens, but about infected cells. So here we have an infected cell. So this could be an infected macrophage or an infected dendritic cell, right? This is going to be infected with a virus, and it's going to display that virus on the outside of the cell. When it displays that virus on the outside of the cell, it's going to bind with a CD8 naive cell and a CD4 naive cell, okay? And those naive cells will then become activated. Once they're activated, the CD4 cell is going to induce co-stimulation to the CD8 cell by secreting some cytokines that are going to tell the CD8 cell to turn into a cytotoxic cell. And that cytotoxic cell is going to create activated cytotoxic T cells and memory T cells. These activated cytotoxic T cells, remember, are going to go around and they're going to hunt or look for any kind of infected cell. And when they do, they're going to secrete perforin and granzyme, and they're going to kill that cell. On the CD4 side, remember that the CD4 is directly activating the CD8 cells, but don't forget that the CD4 cell was also important for activating the B cells. Okay, so it is kind of a link between cell-mediated and humoral immunity in terms of the adaptive defense system. Okay. At the same time, remember that we have to play into some innate immunity here, because the cytokines that the T helper cells are secreting, that are inducing this co-stimulation, that are helping proliferate B cells, is also causing macrophages to begin chomping up more. They also help enhance all of the innate defenses in terms of inflammation. So we have this big connection back to our innate immunity that we also need to make as well. So utilize this flow map to help you understand some more. I am going to be doing a review video, kind of an overview of all of it together. That doesn't go as in depth into every little key player, but it's going to be a nice big overview of connecting all the pieces together. And that video will help if you're still a little unclear on a few things, or if you need to just kind of go over one more time how all the key players work together. Additionally, I'm going to go ahead and upload later this week my flow map. That way you have a big comprehensive view of everything that you really should have on your flow map. And so I'll have that later in the week, but I still want you to make your own, because I think putting pen to paper and really kind of doing that helps review the things that we just talked about. All right, I hope that this video helped. I hope that you're not totally confused. And I hope that you're enjoying immunity. It really is a beautiful, complex system that our body does in order to help keep us safe. So hope you're having a great week. I miss y'all dearly, and I hope to talk to y'all soon. Bye.